Hi, this is Group 2 with Nicole Nguyen, Jennifer Van, and Fiza Nakvi, and the topic of our presentation today is about the effects of different drugs on frog heart rate and contractility.
To start, we'd like to do a quick moment of silence for all the frogs who dedicated their life to science throughout the quarter. Okay, moment over. Introduction and research topic. This experiment analyzes the electrical and mechanical events of a frog heart using ECG and contraction recordings to determine the effects of dantrolene, amidrone, and dopamine on heart rate and contractility. Clinically, dantrolene is a skeletal muscle relaxant that treats disorders that causes increased muscle contractions or spasms, such as malignant hyperthermia. Dopamine is usually used to treat conditions such as cardiovascular shock or congestive heart failure that require increased blood flow, and amidrone is a class 3 antiarrhythmic drug and is used to treat arrhythmia cardiac conditions and mainly slow the heart down. The heart rate, amplitude of contractions, waves, and intervals present in each ECG and contraction recording were compared and used to study cardiac physiology fundamentals. Right, a brief method section. The dissection and preparation of the frog heart was based off the BIPIN 105 lab manual. For the experiment, first the isotonic transducer was calibrated, and then the drugs were dripped onto the heart in the order of dantrolene, amidrone, and dopamine from least to most toxic, using a pipette full of each, and then ECG and contractions were recorded from all of them. Drugs are washed off using Ringer solution in between each treatment, and controls were taken prior to each of them as well. All right, for our setup, the left is a diagram of the isotonic length transducer setup, and the right shows our preamp settings. For the experiment, we used a positive monopolar recording with the positive electrode touching the ventricle. All right, we have two calibration equations for this experiment. This was used for conversion of transducer output voltage in millivolts into the length that the muscle shortened during contraction in millimeters. The left is the calibration equation that was used to calculate the systolic contraction amplitude for the drugs amidrone and dopamine. And on the right, we have a separate calibration equation that was used for calculating the SCA of dantrolene. All right, our sample ECG and contraction recordings. The left one shows when we had separated SVP and T waves, and this is how the ECG recordings appeared for the treatments of amidrone and dopamine. The heart rate was calculated by measuring the RR interval, or the time between each R as shown in the figure, and dividing 60 by that value to report the heart rate in beats per minute to the nearest whole number. The systolic contraction amplitude, SCA, was found by measuring from the start to end of systole, or contraction, in millivolts of the transducer output, and using the calibration equation produced to convert and report SCA as the length of contraction in millimeters. The PR interval is measured from the beginning of the SVP wave, or SV and atrial depolarization, to the beginning of the QRS complex, or ventricular depolarization. It measures the time it takes for the SV and atria to depolarize, for the depolarization to travel through the AVN, the bundle of His, and Purkinje fibers until the beginning of ventricular depolarization, or the QRS complex. The QT interval is the time from the beginning of ventricular depolarization to the end of ventricular repolarization, or the end of the T wave. On the ECG, the QT interval is a measure of the total time it takes for depolarization and repolarization of the ventricles to occur, or a complete action potential. The length of the QT interval corresponds to the action potential duration, so a faster heart rate shortens the length of the QT interval as the time between action potentials and depolarizations is shorter. When the ECG recording produces a combined SVP and T wave, as seen in the figure on the right, PR and QT segments are used instead. This was how the ECG recording appeared when the frog heart was treated with dantrolene. Segments on the ECG measure the portion between two waves where no net electrical activity is occurring, whereas intervals on the ECG measure a particular wave and the length between them. Segments on the ECG measure the portion between the two waves. The QT segment is the time between the end of the QRS complex and the beginning of the T wave, and measures when the ventricles are depolarized in phase 2. The PR segment is the portion between the end of the SVP wave and the beginning of the QRS wave. It represents the time before ventricular depolarization, while the signal from the atria is passed to the ventricles. All right, moving on our results from each treatment, we're going to begin with dantrolene.
So starting off with dantrolene, when dantrolene was applied to the frog heart, there was no significant effect on the heart rate in SCA. For dantrolene, the ECG recording produced had a combined SVPT wave, and so segments were used to measure different portions of the recording. With that being said, the PR and QT segment also showed no significant change, and clinically, dantrolene is typically used as a skeletal muscle relaxant. So dantrolene acts by inhibiting the function of ranitidine receptors, RYR, which are calcium release channels. Dantrolene primarily acts on type 1 ranitidine receptors, RYR1, on the sarcoplasmic reticulum and skeletal muscle cells. It does so by acting as an inhibitor for sarcoplasmic reticulum vesicle release by binding onto RYR1 and inhibiting the channel from opening, which stops the calcium in the sarcoplasmic reticulum from leaving. As calcium is required to trigger muscle contractions, this normally decreases muscle contractions in skeletal muscle. However, dantrolene has been found to have no effect on RER2 in cardiac cells, which is activated by the calcium that enters through the L-type calcium channels that then results in the release of calcium from the sarcoplasmic reticulum. Dantrolene typically has minimal direct effects on the pacemaker activity of the heart because the primary mechanism for depolarization and or contraction of the heart is driven by calcium brought in through RER2 from the sarcoplasmic reticulum, which dantrolene does not modulate. So because of that, there is no significant effect in the heart rate, no chronotropic effect, and also no significant change to segment length, as mentioned before. Additionally, the use of RER2 instead of RER1 in cardiac cells also means dantrolene has no significant effect on the cross bridges formed through calcium binding to troponin, so there is no inotropic effect as well, which means that there is no change in contraction size, which can be seen by the minimal change in SCA.
So amiodarone is typically used to manage and stabilize cardiac arrhythmias, and it's usually used to decrease the heart rate. And our overall trend once amiodarone was applied to the heart was that the heart rate and SCA values decreased while the intervals increased. Okay, so for the mechanism of amiodarone can block calcium channels, and by doing so, that will decrease the rate of depolarization. So more specifically, it's decreasing the calcium ions in the cardiomyocyte myoplasm, and that's what leads to a negative inotropic effect because amiodarone is effectively interacting with potassium and calcium channels while also partially blocking the adrenoreceptors. So what this means is that with the potassium channel blocked, it stops the heart from being able to repolarize due to the fact that potassium won't be able to go out, and the reuptaking process of the calcium into the SLR will also be decreasing due to the potassium channel blockage. And additionally, amiodarone is going to be blocking the calcium channels, so without an influx of calcium going into the cytosol, there won't be many cross-bridge formations, and that's going to lead to a decrease in power strokes and therefore a decrease in contractility. And now the negative chronotropic effect is caused due to the potassium channel blockage. So again, since potassium won't be leaving, the refractory period will be longer, and that means that the firing of the action potentials will also be longer. And a slower rate of action potentials being fired means that the heart rate is essentially going to be beating slower. The blockage of both the potassium and calcium channels are also going to be slowing down the conduction velocity within both the SA and AV nodes. More specifically, it's the L2 type calcium channels being blocked, which is why the rate of depolarization is decreasing. Action potentials also go through different phases before it hits the refractory period, and in phase 2, the L-type calcium channels are usually activated, which allows for lots of calcium to be coming in throughout this longer phase while potassium leaves. But Since the depolarization phase is inconsistent and unstable, it's causing the refractory period to become longer, and that is also why the QT interval is prolonged once amiodarone is applied to the heart. Dopamine is typically used to increase blood flow for conditions such as heart failure, and our overall trend once we applied dopamine to the heart increased our heart rate and SCA values while decreasing our PR and QT intervals. So now focusing on the dopamine mechanism, what's basically happening is dopamine allows more calcium to enter into the cytosol, and that's going to make the SERCA2 pump more active. Therefore, it's going to separate into the SR and remove it from around the myofilaments more quickly. And since the calcium is being rid of at a faster rate, it's being moved back into the SR, which is going to cause a shorter contraction duration and a faster heart rate. Additionally, following the mechanism, the binding to the beta-1 receptor will increase the influx of calcium, which is also going to result in the resting memory potential to be increased and therefore reach threshold quickly. The beta-1 receptor is also going to activate the G protein, which is a GS transduction pathway. The GS alpha will activate adenylate cyclase, which converts ATP into cyclic AMP, and that's going to activate pKa. Now that there's a protein kinase that can go around and phosphorylate things such as the L-type calcium channels, the L-type calcium channels will become more active and lower their threshold without needing to be depolarized much, which leads to an even larger influx of calcium heading to the cytosol. pKa essentially increases funny channels as well, which is going to increase the rate of our depolarization. This means that depolarization phase will occur a lot more quickly and the action potentials will be fired faster and cause an increase in the heart rate. Since the beta-1 receptor is increasing cyclic AMP production and L-type calcium channel activity, it means that there's a faster conduction velocity as well, which is going to be increasing the contractility, and that's therefore why our SCA value increased. An influx in calcium will also lead to an increase in cross-bridge formation and power strokes, which is going to cause a positive ionotropic effect similar to amiodarone.
So, in conclusion, just to summarize our results, dantrolene didn't significantly change heart rate, segments, and SCA. Amiodarone decreased heart rate and SCA, but increased the length of the PR and QT intervals. And dopamine increased heart rate and SCA, but decreased the length of the PR and QT intervals. In relation to how this research is important, since frogs and humans share similar physiology, the effects of the drugs tested should operate in humans similarly, and this is further supported by papers referenced in our experiment. Additionally, the study and experimentation of these drugs could encourage more use in cardiac-related situations such as heart failure or arrhythmia that we've mentioned before, with the added possibility of learning about other effects on other mechanisms that could treat other conditions that we don't currently know about. And with that, that is the end of our presentation. Thank you for watching.